system. A pair of kidneys. That diagram you are done, all of you?
Yes,
ma'am. That is done. ये इसको मोड क्यों चेंज नहीं हो रहा आपके साथ? मोड क्यों चेंज नहीं हो रहा? Now the parts of the excretory system, the diagram already you have drawn. What are the parts? A pair of kidneys, a pair of ureter, one single urinary bladder, and the urethra. Sphincter muscle. मैं इसको मोड चेंज कर दिया, इसको मोड पे कर easy मोड पे करके रख Now next is what did we learn? What is the major function of the kidneys? What are the function of the kidneys? We had studied, no? Two major function of the kidney. What are you supposed to write? Two functions of the kidney. What are you supposed to write? Filtration of the blood and
Formation of the urine.
that is the same word. Formation of the urine and filtration of the blood. And second function,
Maintenance of
salt and water balance. So always to write that. Filtration of the blood, formation of urine. We are going to understand the process today. Second is maintenance of the salt and the water balance in the blood. Next is we were talking about parts like the ureter. What were the functions of ureter? Carrying the urine from the kidney to the, look into your diagrams and all of you refer to the diagrams as I speak. All of you have finished the diagrams, no? Function of the ureter. What is the function of the ureter?
Carry the urine to the bladder.
From the kidney to the bladder. What was the function of the bladder? Temporary storage of the urine. Urethra and ureter. What is the difference? Urethra and the ureter. Ureter is carrying the urine from the kidney to the bladder, and urethra is releasing the urine out from the body. Now, talking about the process of excretion here. Did we finish with the parts of the nephron? Did we draw the diagram of the nephron?
No, ma'am.
Diagram of the nephron we haven't drawn. So then, write one question there. Parts of the excretory system is over. Write one function there. I have given you, no? Volume three questions. What's volume के देना questions?
Three questions.
कौन से three questions?
Name the blood vessel that carry toxic blood towards the nephron.
I'm going to put my sign in your notebooks. सारा proof में रखती हूँ. बाद में जब marks कम आएगा ना, I'm going to squeeze your bones. Check. Those of you have not done your work. Squeeze your bones का method भी मैं बताऊँगा. So now, पता नहीं. कभी किसी ने बताया नहीं तुमको कैसे करते हैं? So now, homework जिसने आज करके नहीं आया, after the class you will stay back for another 10-15 minutes. You will finish your work, show me or show the coordinator, and then only you will go. Okay. So now, what are the questions? I will just discuss. Tell me what are the questions. I will just discuss. सारे books with your work done. Then only I will put my sign, and that remains from time to time. I will check. Number one, what is the first question I gave you?
Name the blood vessel that carry toxic blood towards the nephron.
Blood vessel that carry toxic blood towards the nephron. What to write? Renal artery. Next.
What type of muscle make up the tubules of excretory system?
What to write? Smooth muscles. Smooth muscles. Number three.
What is the role of sphincter muscle in the urethra?
In the urethra. What to write?
Sphincter muscle play a major role in controlling the flow of urine. They work together to maintain urinary consistency.
Handwriting नहीं है.
Preventing the leakage of urine and allowing for voluntary urination when needed.
इसीलिए कॉपी नहीं करना चाहिए, समझ के लिखना चाहिए. कहाँ से कॉपी किया?
वो handwriting mistake.
अच्छा चलो. Now the first two answers are simple. All of you could write. What is the role of the sphincter muscles? Check. What is the role of the sphincter muscles? Role of the sphincter muscles. What are you supposed to write? Role of the sphincter muscles is to regulate the flow of the urine from the bladder. Regulate the flow of the urine from the bladder, empty the bladder. So this is the specific 1 mark answer that you write. Sphincter muscles can regulate, they can contract, relax from time to time and regulate the flow of urine from the bladder.
To prevent leakage.
And also, because these are voluntary muscles, we can control and to also prevent the leakage of the urine. So that is also another point that we can write.
Regulate the flow of urine and
Regulate the flow of urine from the bladder. Regulate the flow of urine from the bladder. And this is the most important point that you should mention. The second point is to prevent the leakage of the urine because sphincter muscles are voluntary. To regulate the leakage of the urine. Take the fourth question now. Take the fourth question. Write two differences between.
and you give four questions.
और कौन सा है fourth?
What is meant by glomerular?
अच्छा, what is glomerulus? I must repeat it. What is glomerulus? What did we understand that day? What is glomerulus? Write down the answer there, Nika. No, glomerulus is not the cup-shaped structure. What is glomerulus? Glomerulus is the network of blood capillaries. Network of blood capillaries formed by, Liko. Network of blood capillaries formed by the division of network of blood capillaries formed by the division of the afferent arteriole. You write this word. We are going to understand. Know this structure now. Formed by the division of the afferent arteriole. Formed by division of afferent arteriole. Now, before we start the diagram of the nephron, let us take just one more question in that set. Number 5. We can continue. Number 5. Write two differences between the renal artery and renal vein. Two differences between the renal artery and renal vein. Table banao, table bana ke properly likho. Two differences between renal artery, renal vein. I will give you the points. You write down. Write two differences between the renal artery and renal vein. All of you will make a table to write down the differences.
Table.
Renal artery, the first point you write. Renal artery carries blood. For renal artery, the first point carries blood towards the kidney. Carries blood towards the kidney. Renal vein you will have to write. Carries blood away from the kidney. Carries blood away from the kidney. Next point. Renal artery, the blood is toxic with a high concentration of urea, uric acid, etc. Blood is toxic with a high concentration of urea, uric acid, etc. Renal vein, the blood is less toxic
Yes.
less toxic. It is not absolutely pure, but toxicity come in. Renal vein, the blood is less toxic and has lower concentration of uric acid, etc. Urea, uric acid, etc.
यूरिया और यूरिक एसिड के अलावा भी कुछ होता है?
बहुत सारे क्रिएटिनिन है, अमोनियम सॉल्ट्स है, पोटेशियम कंपाउंड्स है. We are going to learn 2-3. वैसे तो कंपोनेंट्स बहुत सारे हैं. Next, the third point, I'm giving you 3 points during for the exam. As per the question, you will answer. Number 3, renal artery. Branch of renal artery, branch of aorta,
BRA.
branch, B-R-A-N-C-H, renal artery, branch of aorta. Aorta spelling किसने पूछा? A-O-R-T-A. ब्रांच ऑफ एओर्टा मतलब वो पेजेस अभी तक वापस देखा ही नहीं.
ब्रांच ऑफ renal artery then branch of
रीनल आर्टरी के पॉइंट्स लिख रहे हो ना तुम. ब्रांच ऑफ एओर्टा, रीनल आर्टरी से ब्रांच ऑफ एओर्टा. Just the other day you had drawn the diagram. किया कि नहीं complete diagram? You were absent. You absent थे? Now वो diagram complete करना. Renal artery, branch of aorta, which divides further. Branch of aorta which divides further to form. Check spelling. To form the efferent arteriole. To form the efferent arteriole. To form the efferent arteriole, comma, and the glomerular capillaries. Efferent arteriole and glomerular capillaries. I'll repeat. We are writing about renal artery, right? We are writing about renal artery. So what is the point you are writing? It is the branch of aorta which divides to form efferent arteriole and glomerular capillaries. Other side for renal vein formed by the union of you will understand in this figure right now. Formed by the union of capillaries and joins with other veins J-O-I-N-S, joins with other veins to form. Veins of the body form what? Veins of the body form what are the big broad veins parallel? अगर मैं aorta की बात कर रही हूँ, then broad veins का नाम क्या है? Vena cava. Joins to form the vena cava. Joins to form the vena cava. V-E-N-A-C-A-V-A, vena cava. Done. Now let us try understanding all these concepts. First of all, the words. First of all, the terms. So now, when we are talking about the structure of the nephron, we are drawing. What is a nephron? It is the structural and the functional unit of the kidneys. This question also you should know very specifically. Define a nephron. What are the parts of a nephron? We are going to study now. Define a nephron. ये question भी आना चाहिए. If you have not written, you have to write it. Define a nephron. What is the definition of a nephron? Structural and functional unit of the kidney. Structural and functional unit of kidney. ये complete कर लो, पहले लिख लो, and then we are going to start understanding the process. What is the nephron? The structural and functional unit of the kidneys.
नहीं बनेगा भाई ऐसा से थोड़ा सा अलग बनेगा चलेगा.
Now let us try understanding the parts of a nephron. What am I drawing? This is the structure of a nephron. And where are these structures present? The other day you had drawn the diagram of the kidneys here. What were the two major regions of a kidney? If I talk about the structure of the kidney here, the structure of the kidney we have come across, that this structure of the kidney that we understand should always have two major regions. What are the two major regions we are talking about? The outer region in the other day in those images also I showed you. The outer regions consist of large number of some bead-shaped structures as if, and this outer region of the kidney is known as the cortex of the kidney, whereas the inner region of the kidney, which is comparatively lighter, known as the medulla of the kidney. So let us start from here now. This particular concept, the other day also I told you, again we are going to understand this. The kidney when cut in a longitudinal section. In a longitudinal section, very clearly we can see that the inner region of the kidney is comparatively lighter in shade, meaning there are a large number of structures present, but they are less dense, less packed. So since they are less dense, less packed, this particular region appears a little bit lighter in shade. In comparison, when we talk about these margins of the kidney, the margins of the kidney appear darker, dense, darker, as if something is packed so much. Then try to understand what are those structures packed so much and what are these granular structures actually there. These granular structures, when magnified, it is seen that as if it is a globular head-like structure. And this globular head-like structure runs down till the medulla in the form of a long tube, and then again coming up and then uniting and finishing up here. So there are some structures here all over the kidney, and in millions they are. So what are these structures? जो एक globular head जैसे structure से शुरू होके एक long tubular structure till the medulla and again coming back to the cortex. So this entire long tubular structure, what are these known as? These are known as the nephrons. Definition क्या लिखना है? Nephrons are the structural and the functional unit of the kidneys. क्यों? Why structural? Because the entire kidney is made up of the same typical structure. Millions, in millions the structure is the same. Functional unit क्यों है? Because all of them are performing the same function, filtration of the blood. So nephrons are known as the structural and the functional unit of the kidneys. Justify, explain, what will you write? Nephrons are known as the structural unit of the kidneys because similar type of structural unit are present in millions in the kidneys, and all of them are performing the same common function of filtration of the blood. So that is your answer that you write. What are the parts now? Let us try labeling each of these parts. So this particular blood vessel, what is the name of this particular blood vessel? Here we now start with the first blood vessel, again the renal artery. That just now you have written down the renal artery. We are comparing two blood vessels now. I compare two blood vessels now, the renal artery and this particular blood vessel. I name it as the renal vein. So two blood vessels, just now you have written down the differences. If it is the renal artery is carrying the blood towards the nephron. This entire structure is the nephron. Now this renal artery, as it is entering the kidney, the renal artery again divides to form into narrow blood vessels. What are these narrow blood vessels known as? These narrow blood vessels are known as the afferent, A-F-F-E-R-E-N-T, afferent arteriole. What are they termed as? Afferent arteriole. Arterioles are always branches of artery. Arteriole. Then we have this afferent arteriole dividing to form a network of blood vessels. What are this network of blood vessels known as? Glomerulus. You had answered just now also one question. What are glomerular capillaries? Or what is glomerulus? What are you supposed to write? Check here now. What is glomerulus? A network of blood capillaries formed by the division of afferent arteriole. Correct? A network of blood capillaries, जिसमें hundreds of blood capillaries are all tangled, resulting in the formation of a ball-like structure as if. So this network of blood capillaries, which are formed by the division of the afferent arteriole, that is the definition for glomerulus. But if I talk about the glomerulus, where is it packed? It is packed in a double-walled cup-shaped structure. So ये double-walled cup-shaped structure को क्या बोलेंगे? What is the name of this? The double-walled cup-shaped structure is known as the Bowman's capsule. So this entire structure, the Bowman's capsule, if I think of what is it made up of, it is nothing but made up of smooth muscles, almost like the rest of the internal parts of the body. But one feature of the double-walled cup-shaped structure to understand is the outer wall. If you see, this is how you should draw the diagram also. The outer wall that we draw with solid lines. Matlab, the outer wall is even and solid. The inner wall, if you check, I have drawn with dotted lines. क्यों? Because the inner wall is porous. बहुत सारे openings है, holes है on the inner wall. So that's the reason we draw the inner wall porous. We draw the inner wall with dotted lines to signify that it is not a even layer, or it is a porous layer. And the inner region is all hollow, just like the intestine, just like the stomach. The inner region is all hollow. So exactly the entire region, right, start from the Bowman's capsule, is an entire hollow structure from here to the end. So now the glomerulus and the Bowman's capsule, together known as what? This is known as the Malpighian body. This is known as the Malpighian body. So what is Malpighian body? What will you answer? Malpighian body is made up of the glomerulus and the Bowman's capsule, which is actually this head-like structure that is seen in the cortex. Then we have the long tubular part, this entire tubular part. Now, this tubular part has got specific names. This tubular part has got specific names, but in your syllabus, they are only mentioning as the first and the second and the third part of the tubule. So I will mention both. If you feel you want to write the specific name, then also you can write, or else you can write down as first, second, third part of the tubule, not a problem at all. So now we talk about that this is the Bowman's capsule, this is the glomerulus, this entire head-like structure, what is it known as? This is known as the Malpighian body. What about the tubular part? The first part, the first region. Now if I talk about this part, the first tubular region, what is this first tubular region known as? The first part of the tubule. This first part of the tubule is known as the PCT. PCT. Now I'll just give you the short form. As I said, if you want to write, you can write, or else this is optional for you. क्योंकि ये सारे names book में नहीं है, but if you want to write, you can write, not a problem. So the first part is known as the proximal convoluted tubule. Now, first part of the tubule of the nephron is why the word proximal? Because it is near to the Malpighian body. Proximal from the word proximity. Proximity का मतलब क्या है? Near. So why the word proximal? Because it is near to the Malpighian body. And why the word convoluted? Because it is coiled, extremely coiled. That's the reason, convoluted. So proximal convoluted tubule, first part of the tubule जो है, उसका नाम है PCT या तो proximal convoluted tubule. I talk about the second part. This is the second part. What is this second part known as? This second part is known as the loop of Henle. After the name of a biologist who had understood the structures, the loop of Henle. The loop of Henle is the second part of the kidney tubule. The second part. Talking about the third region now. This is the third part. Now,
You said that the binding structure controls the
flow, the blood composition. This entire whole loop of Henle plays a very important role when we are going to understand the volume and the composition of the blood. The third part of the tubule, what is the third part of the tubule known as? The third part of the tubule is known as the distal convoluted tubule. What is the meaning of distal? Distal from the word distant. Distant matlab far away from the Malpighian body. Because ये जो structure है, this plays a major important role, a major role when we are now talking about the formation of the urine and the filtration of the blood. आया समझ में the names first of all, glomerulus, Bowman's capsule, together they form Malpighian body. Tubular portion जो है, tubular portion divided into three regions. What are the three regions? Number one, the first part also known as proximal tubule. Second part known as the loop of Henle. Third part known as the distal tubule. And the last region is known as the collecting duct. Let us now try understanding the process. What are the steps in the process of filtration of the blood and what happens? The entire process is based on certain features. Certain features meaning a process known as diffusion is majorly working here. A process known as diffusion majorly works here. So if I now try to understand the basic characteristics, let me now show you that how exactly the entire process of filtration of the blood would happen. So for that, I will think of a single artery, a single blood capillary. I think of a single blood capillary. And what would happen for that single blood capillary? So first I think of the Bowman's capsule. What is the feature? First of all, I say for the Bowman's capsule, the inner wall is hollow, the outer wall is solid. This entire portion of the tubule, I divide, considering that this is perhaps the first part. This is the second part of the tubule. And this is the third part of the tubule. What about the blood vessels? Which is the first blood vessel I am to think of now? Efferent arteriole se start karna hai, sabse important. Look into the diagram and find out efferent and efferent. Isko bolenge efferent arteriole. This is known as the efferent arteriole. We'll have to draw this diagram after some time. I'll give you time. Try to understand first. Now the efferent arteriole. Can you find out a difference between the efferent and efferent? Difference है कोई? Efferent and efferent arteriole के बीच में structurally कोई difference समझ में आ रहा है? Broad है, broad है. And efferent is narrow. Yes or no? So when we are talking about the structures, the efferent and the efferent arteriole, in your book the diagram that is there, this part is not at all understood. All of you will have to draw this diagram. Okay? So as I am explaining, try drawing or else first listen to the explanation, after that I will give you time. There is time, don't worry. So now that I talk about the process, what are the steps? I start with the first vessel, that is efferent arteriole. Efferent arteriole is broad. It is a broad vessel which divides into capillaries. Now I think of only one capillary as it. I think of only one capillary now. Can you tell me what are some two special features of a blood capillary? We have already learned that. Blood capillary के दो special features बताओ. Blood capillary के structural दो properties बताओ, दो characteristics बताओ. Almost like the wall of the villi के दो features क्या है? Almost like the alveolus के दो features क्या है? Same thing.
Ma'am, वो thin layer of
Porous है. Number 2,
thin layer.
एक wall has to be very thin. Now let me just go back to that concept. I am starting with these vessels. Which are these vessels? Which is this vessel I am talking about? Efferent arteriole. This particular vessel is efferent arteriole. What about this vessel? This vessel is efferent arteriole. All of you take a note of these words.
दोनों में फर्क है.
फर्क ये है, पहले स्ट्रक्चरल फर्क समझ में आया. I asked you. This is broad and this is narrow. So I am talking about the first is efferent arteriole, which divides further to form the glomerular capillaries. I right now think of only one capillary. इसमें hundreds of capillaries are there. Function सारे capillaries में same हो रहा है. So I think of only one such capillary and understanding the features of this capillary. Wall is very thin and they have pores. So ये दो चीज अभी याद रखना है to understand the process. The wall of these capillaries, which are these capillaries? Glomerular capillaries. Since it is a part of an artery, the pressure of the blood should be high or low. Arteriole is a division of renal artery. You had written down very clear. The pressure of the blood should be very high. Do you still remember what are the reasons? Do you still remember what are the reasons for the high pressure of the blood in an artery?
To pump the blood to the whole body.
All parts of the body. So, if you remember that, I told you that artery के wall thick होता है and the inner lumen narrow होता है. The blood is flowing in an artery from the heart, so definitely the pressure is high. So I start from here. The pressure of the blood in the efferent arteriole is very high. What is the direction of the blood flow? This way. The direction of the blood flow is this way. What is the composition of the blood? Plasma, salts, iron, cells है. And now that the blood efferent arteriole is carrying, इसके चेहरे में कुछ भी नहीं लिखा हुआ है. Straight इधर देखना. So now that we are talking about the blood that is flowing in the efferent arteriole, high pressure, the blood is toxic. Renal artery का blood जो है, that is always toxic. Why toxic? Because it has got a high concentration of urea, uric acid. The points now just tally what you have written. This blood which is flowing in this glomerular capillaries is at a high pressure. So now since it is at a high pressure and the wall of these capillaries has enough number of pores, the wall is thin. So what will happen? It is expected that some amount of the liquid part of the blood will ooze out, will leak out and start collecting in the cavity there in the depression, in the small cup-shaped structure of the Bowman's capsule. So what am I talking about? What is this process? The blood is running in these capillaries. The pressure of the blood in the capillary very high. What is the other reason artery, blood flowing in an artery is always at a very high pressure? Again, number 2, what is the other reason for the pressure of the blood to be very high here? The broad efferent arteriole and the narrow efferent arterioles. This is one more factor which is adding on to the pressure of the blood. Whatever the blood is coming out, if I understand that at one specific period of time, 2 ml of blood coming out, can the same 2 ml come out? No. Hardly 1 ml can come out or 0.5 ml can come out because the diameter is extremely different. So when we are talking about the efferent arteriole broad, efferent arteriole narrow. So what am I talking about? What are the reasons? It will be asked in your paper. What are the reasons for the pressure of the blood in the glomerular capillaries? What are the reasons you write? Number 1, pressure of the blood in an artery is always at a high pressure because coming from the heart. Number 2, the uneven diameter of the efferent and the efferent arteriole. The uneven diameter of the efferent and the efferent arteriole is one big reason why the pressure of the blood is here more. So just now I told you what are the reasons for the pressure of the high pressure of blood in the glomerular capillaries. These are the 2 points to write. Number 1, since the blood is coming by the way of an artery, the pressure always will be high. And number 2, broad efferent arteriole, narrow efferent arteriole adds on to the pressure. So observe, pressure is high. Now next what? The blood which is running in this glomerular capillaries will start now oozing out. Oozing out which part of the blood part? Oozing out which component of the blood? The plasma, the liquid part. But liquid part ke saath, what are the other substances that starts coming out? If I try understanding, what are the other substances that starts coming out? What is here majorly, or what is this filtrate known as? This particular filtrate is known as the glomerular filtrate. Who is talking? This particular filtrate is known as the glomerular filtrate. What is this particular process known as? The 3 steps that we are going to understand now. The 3 steps. The first step, the first process that we are going to come across would be ultrafiltration. Let us talk about the phases, the steps, the stages of filtration of the blood. I am talking about the first step. What is the first step? The first step is known as ultrafiltration. Where is ultrafiltration happening? Here. Why is it called ultrafiltration? Filtration of the blood at a very high pressure. Where is it happening? Glomerular capillaries. What is it giving rise to? Glomerular filtrate. Now, what is the composition of the glomerular filtrate? So, I already told you this is the first part. Second, what is the composition of the glomerular filtrate? Now, I want to know what is the composition after the process of ultrafiltration. What has happened? What is this glomerular filtrate containing? It contains a huge amount of water. It has got a large amount of sodium. It has got glucose. It has got so much of salts. Is urea there? Hardly. Hardly. So traces of urea, traces of uric acid. So what is the composition of the glomerular filtrate? A huge amount of water. Large amount of sodium, glucose, salts, so much of the proteins. Blood cells will generally not come out, but blood cells, if it comes out, also few WBCs, otherwise nothing. So this is plasma only. So not only the plasma, there is additional amount of the water. So the glomerular filtrate is extremely dilute. So what did I say? This entire process, when the blood is running in the renal, the efferent arteriole, passing by this glomerular capillaries, a huge amount of water, salts, proteins, ions are filtered and collected where? In the cavity or the depression of the Bowman's capsule. But I also mentioned that the entire Bowman's capsule is hollow. So definitely the fluid is now running into this cavity. What is the fluid known as? Glomerular filtrate. What is the composition of the fluid? Water, sodium, glucose. A huge amount of water, always to remember. So now you tell me that if this is efferent arteriole and this is the efferent, and I say that this is the direction of the flow of the blood, is this blood thick in the efferent arteriole?
Yes.
Is the blood thick in the efferent arteriole? Is the blood thick in the efferent arteriole? अगर यहाँ पे ultrafiltration हुआ है, glomerular filtrate बना है, so much of the water has come out. Is the blood which is going in this vessel, यहाँ पे efferent arteriole, is the blood thick?
Yes, ma'am.
Definitely thick.
Yes, ma'am.
The blood is coming this way, filtration हो रहा है, whatever the blood is here, ये capillary blood में से यहाँ पे जो भी blood run हो रहा है, almost 90% of the water is coming out. A process known as ultrafiltration is happening. Why the word ultrafiltration? I told you, filtration of the blood under high pressure. What are the reasons for the pressure? I told you just now. What are the reasons for the pressure? So now the glomerular filtrate and the rough composition this way. Next one, this blood capillaries also we have to think, this is how you are supposed to understand that this blood now which is moving, where, how am I comparing? I am comparing that now what would happen between the tubular part and the capillary? Now what will happen between the tubular part and the capillary surrounding? What will happen between the tubular part and the capillary surrounding? That is what you are supposed to understand in the entire process. So what is the first step we had learned? Ultrafiltration. Ultrafiltration gives rise to this, composition is this, and the fluid is passing through. So in the tubule now, what is there? A large amount of water, sodium salts, glucose. So much of the water is there in the tubule of the nephron. What is the first part of the tubule I mentioned? Remember the names, PCT I am talking about. So this is the first part of the tubule. I am to understand that now that the blood passes by the efferent arteriole and goes in these capillaries. First part of the tubule के आसपास बहुत सारे blood capillaries are wrapping it. पूरा जो tubule है, the entire tubule is wrapped up by a network of blood capillaries. Now what is happening between the tubule and the blood capillary surrounding? So I would say here in this blood, what is the concentration of the components? Water बहुत कम है. It is more and more of urea. It is more and more of urea. It has got so much of uric acid, so much of potassium salt, sodium and glucose just traces में है. Sodium and glucose is just in traces. So now traces मतलब बहुत ही कम है. So try understanding what am I comparing? Abhi in this region I am comparing what is happening in the tubule and what is happening in the blood capillary surrounding. नहीं समझ में आ रहा है? You have to raise questions. It is happening all throughout, one after the other. So first I am talking about this region में क्या हो, because it is in parts. यहाँ पे क्या हो रहा है, यहाँ पे क्या हो रहा है, यहाँ पे क्या हो रहा है. The processes you are learning part by part में you have to write the process. So first हो गया ultrafiltration, सबको आया समझ में? What are you supposed to write? Name of the filtrate, site of the process, why the name of that process. Yeah. After that, I talk about that now that the fluid is passing through in the first part of the tubule, एक लगभग ये है composition of the fluid, very dilute, so much of the water, the body cannot throw out these substances. Majority of the substances are essential. Is water a waste? Why are you talking? तुम्हारा कौन सा biology चल रहा है वहाँ पे? Move apart. इधर आओ. After this, I give you questions. अगर नहीं समझ में आया ना, if you're not able to write. एक sentence I think more than 5 times I am repeating. Now, when we are talking about the fluid in the first part of the tubule, is it dilute? Extremely dilute. Yes. So much of the water I am mentioning, there is so much of the water comparatively in the surrounding blood vessel, water बहुत कम है. What is the blood now having in the nearby blood capillary? The blood is thick. Blood में water बहुत कम है. So much of urea, uric acid है, essential substances much lesser. So the body always starts showing a lot of exchange of substances. So exchange of substances, which process are we to understand? Exchange of substances by a process known as diffusion. Exchange of substances by a process known as diffusion, where we understand that the molecules will be moving from a region of high concentration to a region of low concentration. This is the kidney tubule, this is the parallel blood vessel nearby. So the blood is having very less of water, the blood is concentrated, the volume and the composition of the blood has to be revived just in few minutes, otherwise the body will start showing irregular functions. So how does the body react? Immediately a process of diffusion starts happening. Diffusion of what majorly takes place in the first part of the tubule? A large amount of water starts going back to the blood. A large amount of water starts going back to the blood. A large amount of sodium starts going back to the blood. What is this process known as? This process is known as selective reabsorption. What is this process known as? This process is known as selective reabsorption, which is brought about by a hormone known as ADH. Now, ADH and the role and the source of the target gland we will study later in the next chapter. But as of now, what is the role of ADH? Anti-diuretic hormone जो है, this anti-diuretic hormone is actually responsible for this process. So now, the body is not losing out so much of the water salt ions, and the tubule gives it back to the blood. Now,
water उधर ही जाने तो मैम lose to filter क्या होता है?
Water इधर ही अगर जाना है तो filter क्यों होता है? If filtration would not have happened, then the pressure would not have been uneven between the blood and this, and the urea, uric acid, and all these substances would not have come towards the tubule. This is only for adequate amount of filtration happen for the urea, uric acid. The processes are built up in such a way. So we are talking about this. This is happening in the first part. So this is there. Now next again, now that the fluid comes in the second part. What is the second part? Known as the loop of Henle. Now in the loop of Henle, what's now? In the loop of Henle, what is that process going on? In the loop of Henle also, a huge amount of water reabsorption will happen. In the loop of Henle, when I understand, what happened? Now that we are talking about that this fluid comes down to the loop of Henle, here a large amount of water reabsorption will still go on. By the time the fluid has come down to the loop of Henle, meaning by the time the fluid has come down till here, from the fluid, a huge amount of water has gone back to the blood. So I am actually talking about a process of movement which has happened from the tubule to the blood. Every time from the tubule to the blood, from the tubule to the blood, from the tubule to the blood vessel, every time the movement is happening. Are you clear? So from the first part, what are those substances that has gone back to the blood? A huge amount of water, salt, sodium, glucose. What is the second part? Again, a huge amount of water. So by the end of the loop of Henle, what has happened here in the blood? Yes, the blood has got back enough amount of water now. The blood has got back enough amount of water now, sodium, glucose. Yes, the composition is almost reviving back. What is this entire process known as? This entire process is known as selective reabsorption. What did I say? Selective reabsorption means what? The diffusion of essential substances from the tubule towards the blood. But still, I have to understand that what is happening to these components. What is happening to this component? These components are still there in the blood. They did not come till the tubular part of the nephron. So now that I talk about this urea now here, the uric acid now here, the potassium ions now here, all the poisonous chemical substances now here, they will move from the blood towards the last part of the tubule, that is the third part of the tubule known as the DCT. So the diffusion of these substances does not happen in the PCT or the loop of Henle. Why? You should have asked me that question. Why that does not happen? Why am I not saying that the urea moving this side here? No, that does not happen because the membranes of the PCT and the membranes of the loop of Henle are impermeable. Do you understand the meaning of impermeable? 9th standard में पढ़ा ना, hypotonic, impermeable, permeable. What is the meaning of impermeable? A membrane which does not allow the movement. A membrane which will not allow the movement of certain substances. पूरा period क्या उसका चेहरा देखते हैं? हाँ? I think he deserves a better choice in life. Huh? Now, when we are talking about this particular condition, what am I saying? The PCT and the loop of Henle, इसके बारे में बाद में एनालाइज करेंगे. The PCT and the loop of handling. What is the main process going on there? Selective reabsorption. And the third process I told you is, what is the third process? The third process is known as tubular secretion. What is the third process known as? The third step is tubular secretion. What is tubular secretion? The diffusion of a large amount of urea, uric acid, potassium ions, etc. from the blood towards the tubule. So then here now we are getting an adequate amount of urea and uric acid and potassium ions and all the toxic substances. Now where will this lead to? Now the DCT, that is the third part of the tubule, where will it end up into? Where will it meet? It will meet here in the loop, in the collecting dam. That is the last part of the tubule that we are talking about. So here in the PCT and in the loop of handling, which processes take place? It is the selective reabsorption that takes place. What is meant by selective reabsorption? Movement of essential substances from the tubule towards the blood. And what is tubular secretion? What will you write? What is tubular secretion? Movement of toxic substances, diffusion of toxic substances from the blood towards the tubule. Where does selective reabsorption take place? Selective reabsorption takes place, now that you write your answers, you'll have to mention. Selective reabsorption takes place in the first and the second part of the tubule. Where does tubular secretion take place? In the third part of the tubule. So after all these three steps, now what is the composition here? Here the composition would be just a small amount of water, and then urea, and then uric acid, and then potassium ions, and then some specific chemical substances which are toxic. This is what is the rough composition of the urine. Now, the composition of the urine, when you write, urea, uric acid has to be mentioned, and the amount of water just what is necessary for dissolving all these waste substances. Clear? Now, this urine, what will be the pathway? From the collecting duct, where will it go? Look into the figure. From the collecting duct, it will straight away not go to the bladder. From the collecting duct, where should it go? Refer to the figure and tell me. From the collecting duct, it will go to the right? It will go to the broad end of the ureter. Okay, the ureter, then the bladder, then the urethra. Clear? So now, these three steps, we will take one question, difference between reabsorption and secretion. The parts and the function that I told you. Certain questions again the next class we'll have to take. Let us complete with this. Difference between reabsorption, selective reabsorption and tubular secretion. Difference between selective reabsorption and tubular secretion. Okay, before that tell me. I told you selective reabsorption is because of this hormone ADH. If in a person the secretion of ADH is less, then what will happen? If in a person the secretion of ADH is less than normal, then what will happen? Then what is the role of ADH? I told you. What is the role of ADH? I told you. What is the role of ADH? I told you. मैंने क्या explain किया था? Role of ADH क्या बताया था मैंने?
Transfer of water from
The role of ADH is to bring about the process known as selective reabsorption. Why is selective reabsorption essential?
अच्छा तो क्वेश्चन क्या था मेन?
I asked you. आधा से ज़्यादा क्लास को ये कॉन्सेप्ट समझ में नहीं आया. Am I correct? How many of you have understood this? Tell me. I can understand from your faces. पूरा tangent दिया है. Right? You do not understand because you do not listen. What is your doubt? कौन सा part नहीं समझ में आया? कुछ भी नहीं समझ में आया. Right from the beginning, I told you have to listen carefully. Like the many समझ में आया. Which part is not understood? Ultrafiltration समझ में आया?
Yes.
What is ultrafiltration? आया समझ में? Where does ultrafiltration take place? What did I tell you? In the glomerulus. Ultrafiltration majorly is taking place in the glomerulus, and the glomerular filtrate is getting collected in the Bowman's capsule. Then the fluid runs. You have to understand that the fluid is running all throughout, and as the fluid runs, substances are getting exchanged with the blood, surrounding blood vessel. So first part में कौन से substances are getting exchanged? Water is moving from way to way in the first part. Compare this now. Water is moving from way to way, blood to tubule or tubule to blood. Sodium is moving from way to way, tubule to blood. Glucose is moving from way to way, tubule to blood. I go to the second part. Second part, that is the loop of Handley में सबसे important diffusion कौन से substance का होता है? Water. Water is maximally diffusing from the loop of Handley to the nearby blood capillary. This is what is known as selective reabsorption, जहाँ पे all the essential substances, water, sodium, glucose, are moving from the tubule towards the blood.
Blood vessel और loop of Handley एक ही है.
Blood vessel and loop of Handley एक कहाँ है? These are the blood vessels which are formed by the division of efferent arteriole. And loop of Handley is the kidney का tubule है. This is tubule of the nephron. My question is, I asked you, अगर किसी के body में ADH का deficiency होगा, then can you tell me if you have understood? You should be able to answer this.
वो process slow हो जाएगा.
Reabsorption will be very slow, less. Then what will be the problem? The person suffers from a condition known as diabetes insipidus. Where a large amount of urine. What is the normal amount of urine a person should release out? 1.5, 2 liters, maximally 3 liters. Maximally 3 liters. So now if I talk about this glomerular filtrate, are you aware how much of the glomerular filtrate per day? 180 liters of glomerular filtrate per day. कितना? उसमें से how much of the urine loss? Maximum 2.5 to 3 liters. So that is the reabsorption we are talking about. Almost 98% of the water salts reabsorbed back. You will be asked questions. What is the significance of reabsorption in level 2, right? Reabsorption is only to bring about the composition, maintain the composition and volume of the blood. So that part we will answer later. Check, read from the volume, find out whether these 3 steps are clear. The last part, that is hemodialysis, that 2-3 questions left, we will have to complete in the next part. And the drawing of the diagram. Diagram drawing I'll tell you next time. हो गया complete?
Diagram draw तो कर दिया.
Complete हो गया?
क्या क्वेश्चन करोगे?
Diagram drawing I'll tell you. इसमें क्या करोगे? प्रिंसिपल को क्या वो डार्क करो. डार्क कैसे स्लाइड करना है? Diagram drawing में जो पूरा tubule है ना, इसको थोड़ा डार्क करो. That is how you should draw the diagram. What you are doing? Diagram में draw केवल
भाई सारे कॉप पाजी. वो पुत्तर ने क्या ही कर. ओए डरे हुए. अभी भेज बाद में भेजता नहीं है. तो अब कैसे है? ये लास्ट सब तक निकलेंगे. अभी